Hello, friends. I'm Carol Deppy. I'm chair of the board of directors of the Open Source Seed Initiative. On behalf of the Aussie board, I welcome you to our second webinar. Aussie, as we call it, is an organization that is fighting for the rights of farmers, gardeners, and plant breeders everywhere to have full access and rights to the seed we need. Rights to plant, grow, save seed, share, sell, breed with it, and use it however we want. We do this by working directly with plant breeders who pledge one or more new varieties they have created into our program. Thereafter, seed of their variety is distributed through cooperating seed companies with the Aussie Pledge, which says, you have the freedom to use these Aussie Pledge seeds in any way you choose. In return, you pledge not to restrict others' use of these seeds or their derivatives by patents or other means, and to include this pledge with any transfer of these seeds or their derivatives. There are now more than 500 Aussie Pledge varieties being sold by more than 70 Aussie-associated partner seed companies. For more information about Aussie or these varieties, check out our website at osseeds.org. Today's webinar is Combining Cucurbits for Powdery Mildew Resistance and More. The conversation between Michael Mazourek and Edmund Frost, representing one of the few collaborators between a university plant breeder and an independent farmer breeder, each bringing their own unique perspective to their work. Their work is on disease resistance and local adaptation in cucurbits. It often involves wide crosses rare in the mainstream plant breeding world. Michael Mazourek is on the vegetable breeding faculty at Cornell, where he breeds peas, beans, squash, cucumbers, and peppers. He is also a pro bono co-founder of Row 7 Seeds. Edmund Frost is a freelance plant breeder and vegetable seed grower in Virginia. His work focuses on cucurbit crops and downy mildew resistance. He is also a co-founder of Commonwealth Seed Growers. For more on the work of Michael and Edmund, or on Row 7 Seeds or Commonwealth Seed Growers, check out the links in the program notes. I'll now turn these proceedings over to our intrepid moderator, Rachel Holtengren.
Thanks, Kara. We'll be hearing a 45-minute presentation from Michael and Edmund, and then we'll have about 45 minutes for questions and answers. So I'll ask that folks hold their questions until we get to the Q&A. Although if you have any questions that you'd like to share in the chat, I can keep track of those and then make sure that they get brought up at the end of the presentation. So Michael and Edmund, over to you.
Thanks, Rachel. It's cool to be able to chat with you all this afternoon or this evening or wherever time it is where you're at. So Edmund and I, we're gonna talk back and forth. We have a couple different stories to weave together, and then would really look forward to chatting with you all in the Q&A. We're gonna touch on some of the things that excite us about cucurbits, but there's oh so much more we can to talk about well, we like talking of cucurbits, let's just put it that way, right? So one of the first things I wanted to talk about is downy mildew resistance in cucurbits. This is how Edmund and I first connected, I believe, and started an evolving collaboration. But so there's a lot of downy mildews, the basil downy mildew, hops downy mildew, sunflower downy mildew, like everything has its own downy mildew. There are these oomycete pathogens, which is like a fungus, but a little different in how they can sexually reproduce and evolve. While we had great downy mildew resistance in cucumber for decades as part of some older breeding efforts, when there was a resistance breaking strain of downy mildew, it had nothing to hold us back. It was not still really known anymore to plant pathologists, to plant breeders, to extension, all the diagnostic people. This was something that they was kind of out of sight, out of mind for decades. So when it popped up back in the US on the East Coast, about 2004, 2005, that really was devastating to the industry. Appears to overwinter in Florida, move up the East Coast every summer as plantings move further north. Also, there seems to be an epicenter around the Michigan, Ontario area that moves in. It's also global. It's wherever it's kind of humid and they're growing cucurbits, cucumbers especially, it just takes off. So there was a big scramble. There's lots of scientific articles about people reacting to this. What is this? What do we do? How does this help us learn about how to deal with future plant pandemics, right? The symptoms on cucumber are pretty easy to recognize with and pretty much any place humid globally. You grow cucumbers, you'll see this. It has this patchwork quilt appearance where these polygons of yellow and brown is really diagnostic, and it tends to, unchecked, just will rip through a field. So here's a trial in North Carolina with the great collaborators down there in the photo credit. But so what you're seeing, if you're on the audio version, is there's some plants that's going to get some mild symptoms in the leaves, and then there's like 10 days later, they are wrecked. It is just brown and crispy. The plants are gone, right? So it really does a number. And so the spores land, they'll go through their life cycles. They have alternating wet and dry, and then they're airborne and then are blown to the next field. And so that airborne dispersal combined with the sexual mating approach for them to evolve and share genes makes it really something to contend with. At the least, it doesn't overwinter really, and it is restricted by the crop type. Right. So for me, this was like one of the challenges when I first joined the faculty a couple of years later, and I was trying to figure out how to do this. At the time, I was the second to last public sector cucumber breeder left. I am currently the last public sector cucumber breeder left I know of that's really focused there on the plant breeding. So, it's like, we're able to do some things to help, but my goodness, there's almost no one to do anything on the public sector side. Not Aussie related comments later on there, right? But so I tend to be able to make some really fast progress in plant breeding, but I want to unpack that a little. I usually don't unpack that much, right? And so one of those reasons is that I am part of this legacy of seed being saved and archived at Cornell. So I am able to do some things faster than some in some of the breeding spaces, but I'm able to do that because I have this fantastic set of germplasm to work with and publications to give me the roadmaps of what people did. So we knew Henry Munger, Molly John, etc., had left behind this Marketmore 97 cucumber that was the pinnacle of disease resistance as of 1997. It was when they were breeding for Downy Mildew resistance. Or with those genetics still in the background, even though it wasn't a big problem. And so I was able to start working with that, and we're looking to what to combine it with. Michael Gloss, a farmer breeder in the program, was able to give me a great tip that there's this hybrid in Cook's Garden, Ivory Queen, that was this white pickling cucumber. It was not normal to a lot of people, but there were some growers that found it was giving them some degree of protection with resistance. So we combined these two moderate sources of resistance for the backgrounds where, who knows, Marketmore 97, we do Ivory Queen, we still don't quite. And we put these populations out in the field. There's a lot of ways you can work for breeding for disease resistance with a lot of ratings, a lot of data collection. My mind doesn't work that way. We walk the fields at the end of the season. and just look for the survivors. So here's a field of cucumbers where they're all wrecked with downy mildew. Some are just dead and done. Some are struggling and holding on. Some, I have one indicated, has got a pink flag in it, and it's still alive and trucking at the end of the season. It goes right up until frost for us. So this was part of our idea of how can we help the plants evolve like the pathogen is evolving, right? So it's one of the problems with a lot of cultivars and how we keep them as varieties in the stasis is that the world around us, climate or ecology or people or cultivation methods or like everything's still changing. So how can we help set up a system for varieties to evolve, right? So a lot of you are probably interested in open pollinated cultivars and their capacity for that. We were taking kind of an accelerated approach toward that with our greenhouse. So I just go at the end of the season, what plant makes good cucumbers, what plant is still alive and producing. We take cuttings of those because we don't want pollen from all the dead plants mixed in. Cucumbers root so well. So we just take a cutting, put it in a pot of wet soil. We used to just put like a bag over the top, a clear bag. They root within a couple weeks, and then you're often going survival of the fittest for the next generation. For us, it's in the greenhouse. We have a better method now. I had an undergraduate student that I didn't describe this well enough. So she did this approach her way. It took these long, like 18-inch, 2-foot long cucumber whips from the ends of the plant. I was like, oh gosh. And we just put them in these big, like Tupperware clear plastic boxes instead of these bags. They wouldn't fit. They almost all took. So we have like a 50% take rate, kind of depends if it's getting cool at the end of the year. I feel like 100% take rate. We're thinking it's because these long whip she had. Maybe it's because the apical meristem that has all its hormones was just very far separated from the zone we wanted to root. So perhaps that was what was doing it. But so anyway, so the idea here is it's very much a evolutionary survival of the fittest. The survivors are what's intermating, and we're intermating to continue driving the population forward. And talk more about that. But actually, the expert in a lot of the other ways to do it is Edmund, and I don't want to take into his time. The very humble world expert on downy mildew resistance breeding in cucurbits. He will tell you otherwise, but I'm here to give my 2 cents there. So one of the things here is we have I'm showing 2 panels of on the top here, 2 cucumber varieties that are super resistant, one more than the other. So we have this DMR 264 and DMR 401. It's the breeding lines, it's the plots when we first recognize them as having potential. You can see they have potential, I hope, here if you compare them at the bottom 2 panels. So one is straight 8, where you see a couple little brown wisps where the plants were and when downy mildew moved in, and they are gone now. Also, you see a variety that was labeled as resistant at the time. It is not much better than the susceptible control. Right? So a couple things about these 2 varieties. They were developed with graduate students. Bill Holdsworth was the lead and Lauren Brzezowski was the lead. They've taken it further into pickling cucumbers, etc. But these were varieties we released really quickly. They weren't the best we could do. They weren't, we continue to make improvements, release iteratively. Some people ask, when do you know when a variety is finished? Some people say it's never finished. I will say, as soon as you can share it with someone that needs it. Right? So luckily, I had a way to share these very quickly with the public through Edmund's seed distribution efforts. And so first DMR 264, then a couple years later, DMR 401. And we have a couple other new generations that should be out in the world in a year, maybe 2. So that'll be a way to continually sharing with growers immediately the best of what we have to offer. Right? And so I think that's really important for how I approach what I do and how I do it is I want it to be a benefit to people. And even if it's if it could use more years of trialing, if they're desperate to grow a crop, why not let them be doing the trialing as they grow? Don't plant the whole farm to it, of course, but you can start trying to see if it works for you. The other important thing of that is by releasing it commercially for utility patents, two of the hurdles to cross or ways that a utility patent would not get granted is if something's already in the public knowledge. So here's a couple publications, and Rachel, if people are interested, we'll have them around. Rachel might have it in the transcript, where we published on what we did, so it's out in the public space. And the other thing with utility patents is the first to commercialize. So Edmund has a quick delivery approach for these genetics. It is really helping to keep them in the public space by getting them out in the world as commercial cultivars, which means that there can be no utility patents on these genetics because it's public and has been commercialized. So it's old news when it comes to patents. Right? And if you know me, you also know that next I'm thinking about squash, but Edmund, as I said, has surpassed me at Downy Mildew resistance breeding. So I'll turn it over to him. I will note though, in that he does have a slight advantage, which was the disadvantage to the growers around his area, is that he gets Downy Mildew a little earlier and a little bit more consistently than we do in New York. However, it's not to take away from man's skills.
Thanks, Michael. All right, I'm going to share screen as well. So I've been growing seeds in Virginia since 2008, and I always just had an affinity for cucurbit crops, just like the way they grow. Not sure what it is. But I noticed within the first, actually not really the first year, but the second year, started really noticing the downy mildew problem, and just that, I had these seed crops that that were not working well. The leaves were all dying. This is a picture in 2010 of a seed crop, cucumber seed crop that it set fruit and was starting to mature the fruit, but then the leaves all died. So, that wasn't a very productive seed crop there. Probably got a little bit out of it. So that's me. There's a picture of me with a tour kind of thinking about what the hell happened to this crop. So the next thing that happened was, I had an interest in figuring out something better. I didn't want to keep growing these crops that didn't work. And I was learning about downy mildew. And then I went to an Organic Seed Alliance conference and went to a presentation about variety trialing. And actually, I think I met Michael at that conference also. So started thinking about doing variety trials to find seed stocks that might work better. So I started doing that. Actually, 2012, 2013, I did some small variety trials, and then I got a SARE grant in 2014 to do a bigger trial for cucumbers, winter squash, and melon. I did more than I should have done in that trial, but got a lot of information. In one of these pictures here, you can see that the winter squash trial, you can see some squash around the edge that has really died back from downy mildew. The cucumber picture is less far along. The downy mildew hasn't really hit yet. But I tried something like 30 cucumber varieties in that trial, and I tried, 20 squash varieties and 20 melon varieties. And so I had all kinds of questions about how to do this, how to lay out a trial, what to evaluate. And I was always able to call Michael and just ask the questions and be able to get it done right. So that, at that point, we were really starting a collaboration that was valuable to me. And what was valuable to Michael was that I'm in Virginia. And so I was able to trial a lot of his material under conditions with heavier downy mildew and give feedback. And this included the DMR 264 variety, and then a little bit later on, the DMR 401 variety. And let's see. The DMR 401, if you saw Michael's picture, it looks like it's less resistant, but actually it's more productive. So in some ways, it's putting its energy into the fruit. And that's a really, really notable variety. I think has the best resistance out there. So in this trial, I'm going to mostly focus on cucumbers. I was able to identify a handful of seed stocks that did well in the trial. And I was looking at both disease ratings of the leaves. I would go through every week and rate the plants, but I was also looking for productivity because plants that do well in terms of the leaves looking good, but don't produce much fruit, it's not really what I'm looking for. So to some extent, you can kind of rate the productivity, rate the downy mildew resistance by looking at the productivity. So I found a number of standouts. There was actually several Chinese cucumbers that were standouts, in addition to Michael's DMR 264, and a couple other strains that he gave me. And that trial was not only, it was not only a chance to look at the performance of Michael's stuff, but it was a chance for me to find parents for breeding work that I started. And so we're going to look at some of that. This is a trial a few years later in 2018, another cucumber trial. You can see in the foreground, there's some entries that are looking pretty crispy, and some that are looking a little bit better. And now we're just going to look at a few sort of close-ups. This is a picture of Marketmore 76, which is standard slicer. It used to be, listed as a downy mildew resistant variety. And this is a picture of it looking mostly dead. This is at the end of August in this 2018 trial. This is DMR 264 looking very alive. DMR 401 looking almost as alive as the DMR 264, but more productive. This is another line from Michael. This was, I don't think we continued working with this one. This is another non-resistant cucumber called Sumter, mostly dead here. This is a Cucumis anguria plant. It's called West Indian gherkin that I put in the trial. It's very, very downy resistant. It's, something to consider. And then this here is a picture of the variety that I started working on, which is, this is the F3 that I entered into the 2018 trial here. And it's doing, doing pretty good in this picture. I've continued to work on the fruit shape and do more selection, which you'll see. So that's what some of the fruits look like now. It's a picture of some of the fruits going for the pickler shape. I don't have the best picture of it, unfortunately, but so I just want to show you the parents. So there's 3 parents to this variety. I'll start with this one actually. I got this from the Grin system. I actually got quite a number of seed stocks from the Grin system. And I was looking at research that other people had done. I'm forgetting the name of the person that put out this study, but someone had evaluated, a professor from North Carolina had evaluated maybe hundreds of accessions from the GRIN system for downy mildew resistance. So I got information from that and I took some guesses, and this was one that I really liked. It did well for downy mildew resistance, productivity, and also bacterial wilt resistance, which I've been looking at too. You can see the Chinese cucumber fruit shape there. And this is the same 2018 trial here. It looks pretty good, but you know, it's definitely got downy. The other parent is a cucumber from the Philippines that has good pickling quality, also moderate resistance. And then the third one is an American pickling cucumber called Homemade Pickles that, you know, not resistant at all, but maybe did a little better than some of the other ones. And so I actually crossed the Chinese cucumber and the cucumber from the Philippines first, and I did a little bit of selection with that. There was actually really heavy cucumber beetle pressure to select in. So I was happy to find a plant that did well in that situation, cross that to Homemade Pickles, and then started selecting. And here's that F3 again. Already have, you know, some good resistance and productivity. I have taken the approach of, you know, every year since then, every year starting in 2017, really growing that population, looking at the individual plants, and taking notes on flags that are each plant gets a flag. So I take notes about the yields and about fruit characteristics. I taste the fruits. So I look for my favorite plants, and then I wasn't really set up to do the propagation of the best plants the way Michael is, but I have a longer season. So my approach has been to, you know, harvest for a week, for a couple weeks, and start identifying my favorite plants, and then self-pollinate those, or sometimes cross my favorite ones to each other. I could just self-pollinate all of them, but I like to cut down the work a little bit. So that's what I've been doing every year. This is 2018. This is the 2019 version. I actually got a grant from Organic Farming Research Foundation for a couple years of this work. Something else to note is that it's possible I'm able to grow 2 generations of cucumbers in a year, but the first generation, I can't really look at downy mildew. So it can work to sort of mix up the genetics or advance a population, but not really to do the downy mildew selection. You can also do bacterial wilt selection in the early generation. So I did that in 2018, but after that, I started, I asked a grower in Hawaii who was actually on this workshop, Jay Boss, to do winter increases for me, because I'd have a few fruits from my favorite plants that had underdeveloped seeds, and what he did was grow those, harvest seeds over the winter, send me back the seed, you know, a good quantity of seeds to be able to work with the next year. This is the 2021 pickler breeding trial out of that same population. I've also, a slicer line came out of it as well. There's a picture of that. And I think it's definitely, just one last thing. This picture, if you look on the left of this picture here, this is a variety trial in 2021 that includes a lot of new lines from Cornell, including a lot of picklers. So 2020 and 2021, Michael and I have been continuing to collaborate on trials and sending material back and forth to each other. And I think I'll leave it there for a minute, pass it back to Michael.
Oh man, I think in this is kind of, as we mentioned a little bit, the the collaboration part is, I think there's a lot of what we do that's probably going to be really similar. It's kind of going to be how a lot of public sector breeding work happens, where there's only a few university public sector plant breeders left in many cases. For vegetables in the Northeast, there's 3 of us. So you're all set as long as you like squash and kale and cucumbers. But so there's some stability with the seed archives we have. There's some technical insight we have. And that's the thing we can share with people like Edmund, who are the other, I think, public sector plant breeders, public, private, it's kind of both, doing the public's work for sure. But with insight that is at least as impactful and valuable, where we can share back and forth. Edmund is working with genetics that are very different than mine, which is so important because if I was just doing mine and then we get the next resistance breaking strain, we're back to start. So since Edmund is working on alternatives, it's not like, which one of us has like the better one, et cetera. It's like, yeah, you need you need it all, and hopefully maybe 3 or 4 more people having some alternatives. So it's I think it is the type of working together that I'm glad to do more of for sure, and I hope there's some other people that are working on it too. So I wanted to have a shorter vignette about some squash, but squash is really just a way for me to be thinking about diversity in crops and how that is scattered across the globe. And then how do you recapture some of that diversity? I'll be maybe talking about things in a very kind of component sort of way. It is not that I don't love my plants. It's kind of love-hate sometimes, but it's not to say there's not a personal relationship with them and there's not that appreciation at that level, but if we want to talk about the genes and the genetics, it gets rather component-ish, right? And so in that, one of the things it's, so how does biodiversity change? How's our genetic reservoir of all the natural genes in a crop? How is it scattered across the world? How can we bring it back together? So a little bit about squash. So maybe some new thoughts in an area. Maybe a couple of you have heard me talk before, but there's many species of squash. They are grown throughout the world in many different environments for many different uses. Jack-o'-lanterns and acorns and delicatas and the Cucurbit pepo, all the squash with those corky wine bottle cork-looking stems, the maximas, Rouge Viftitowns, buttercups, cabochas, the giant world record ones, and the tropical pumpkins, which also includes butternut, that are just resistant seemingly to everything that, alas, the pepos and maximas are not. Maxima especially is so delicious and so odd stacked against it. Right? So as we're looking at the diversity in this Cucurbita genus and there's other species with varying levels of difficulty, they can all be crossed together, but they all share a couple issues where there's this, many crops had this Cucurbita, maybe a little bit more than some, through a big genetic bottleneck. And so we lost a lot of diversity, things like mass extinctions or something, or a few plants in a founder effect. We don't think we have a perfect idea of what that was, but there is somewhere in the development of the genus and pre-humans, a lot of diversity was lost. In terms of genetic diversity, there's not a ton in Cucurbita. You can self-pollinate them. There's no inbreeding depression, which means you can't make things worse for the plant. So self all you want. Success PM is a variety from Molly, my predecessor that's out in the world. It's an F22, 22 generations of selfing. It's still, there's usually rumors of very little heterosis, right? So that's kind of an intriguing thing. So there are though ways to recover some vigor, get some interesting traits, right? So there's an interspecifics, and so popularized in some schools of thought in Oregon. Tetsukabuto, which is a rootstock, really disease resistant for grafting. I have tasted it at a Culinary Breeding Network showcase. It was so delicious. We can't grow it that way in New York. We've tried. But it's an interspecific squash. It allows you to take what diversity that's left and bring it together. Similar thing, Dick Robinson made the cross in the 80s. It was foundational to what became honey nut. That also, butternut by buttercup. That's what gives it kind of that nice starchiness. He was, I think, looking for some more provitamin A at the time. Right? So to help us figure out, so how can we best look at these? Edmund can do great trials. They are beautiful if you're seeing the photos. They're so gorgeous. But still, even Edmund probably will not be able to trial all 1,300 Cucurbita accessions in our germplasm bank. And so we used the genomic approach where we did a genotyping, not really sequencing the genomes, although we're working on that next, but a way to fingerprint the genomes of everything that was available in the USDA's germplasm bank. So I'm showing a map with the 3 species and the countries of the world they were derived from. But keep in mind, they were all derived from Central, South America, up into like Southeastern US is kind of their range. So they're collected from across the globe, but they're all originally from one part of the globe, where all the heroic domestication first happened before all the other cultures that met them co-evolved with them. So as we look at the genetic diversity, everything that's there, and it's separated by species. So I have just a couple plots here. So some rainbows. So if you were to look on the left side, so there's these rainbow zigzaggedy plots. These are structure plots. So for each species, there's different colors that represent how much of the genome basically they have in common with each other and what part of the genome. So there's yellow that's spread across multiple groups that are on the x-axis. We've improved these further. We've got some more groups. So in Cucurbit people, there's now about 10 groups we've been able to resolve. This will be published soon. Chris Hernandez, brilliant graduate student, I think about to be my colleague in the squash space too. I'm very lucky to have some former students as colleagues in the space, even our humble moderator. I found that in the lucky to be with group. Right. So, right. And so what these are representing is, so you see which are the groups that are genetically related. So what are the branches on the family tree? And there's 6 to 10 now that we can see. And we can also do a plot that separates that out in 2 dimensions, these Pca plots, where I can talk to you about some of the genetic distance things going on. But more importantly, what you can see is there's different colored, there's blobs of different colored dots coming together. That represents all the different geographies where the plants were from. So what we're seeing is there are, as we think about market class, or you think about what country something is commonly grown in, that's a really good predictor of the genetics of it. And so on one hand, if you're thinking about breeding and trying to think about how to get genetic diversity back into a very non-genetically diverse crop, at least I can tell you for Cucurbita, for squash, crossing between countries and market class is something people rarely do in the commercial sector, where you want to make quickly improve your latest hybrid to get next in the market, where you're doing more open wide crosses. These are the crosses you can do between species or just between market classes. You'll get some pretty good genetic variation in the population you'll have. Some of our greatest creations have come from that approach before we really knew we noticed we were doing it, but we didn't know there was any scientific backing for it. Now we're starting to see it, right? What we're bringing together from across the globe is also an interesting thing to think about what it means to conserve genetic diversity, what it means to work with different crops from around the world, how these ex situ conservations for genetic diversity. And there's something there, we're digging into this more, but if you think where a crop was first domesticated, it had all the genes, right? And then as those got moved across the globe, they're taking different subsets of it. But both are continuing to change. So as people grow, cultivate, interact with the plant in all the anthropogenic ways, you're bottlenecking the genetic diversity as you're trying to make it better, right? And so in doing that, you're losing some of the variation that was once there, but you still have it if you find the seed that ventured across the world to other areas where they had different goals, different conceptions of what that plant should be to their culture. And so they kept different subsets of the genetic diversity. And so it's a space I'm really interested in lately, both in terms of kind of breeding progress and kind of what it means for beyond the center of diversity being the archive of all of the variation of a crop, to looking how the globe has become the archive of a lot of the variation of a crop. I have a lot more questions than answers there, but that is a space that we're really excited to think about. If people want to geek out in the Q&A, we can also talk about the allotetraploid origins of the Cucurbit species, genus, etc. But I'd like to hand it back over to Edmund before that. Can't hear you, Edmund. Oh.
Okay, thanks. Yeah. Yeah, that was really interesting. I didn't fully understand the graphs, but I'm gonna look at them some more later. So yeah, that reminded me to talk about that I want to talk about Cucurbita machata. So that's the squash species that does the best in our region and in a lot of the a lot of warmer parts of the world. And I've been doing a lot of work with it, as much as cucumbers, really. You can see in the picture that I have up now, I mentioned the cucumber variety trial on the left. There's a melon breeding trial in the middle. And then on the right, there's a squash breeding trial. You can see some orange flags that, that have names on them. And then, it's not, okay. This is a breeding trial from 2020. Each flag is a different plant. And, notice like the cucumbers, how I keep them trained separately. Learn that from Michael, because otherwise it just gets too confusing. Something that struck me about machatas lately, so about my machata breeding work, the first thing that I did was I crossed Waltham butternut with a Seminole pumpkin, and that was 10 years ago, and then started selecting that for downy mildew resistance. I got downy mildew resistance in pretty quickly, but then was selecting for fruit shape and eating quality and yields and keeping quality. And I think it's a good variety in a lot of ways, but it's I don't know. It's an enigma, and the Cucurbita machata and this selection project continues to surprise me in terms of. I think there's been times when I've thought I've been making a great selection, and maybe it wasn't the best direction to go. And I think the biggest drawback of the variety that I've bred is called South Anna, named after the river that is near here. It's kind of indeterminate. So, you want to go through and harvest it twice, which is not necessarily the most desirable trait for butternuts, but, that is what I do. And so that was, was working for me. It's got great eating quality and downy mildew resistance. And I'm still selecting for that stuff. But then I took that and I started crossing it to other stuff. And actually Jay Boss, again, did some of these crosses for me. there's a squash from Guatemala that we crossed it to that ended up having really good keeping quality. There's a Chinese pumpkin that I crossed it to. That one, the straight cross of those two was a really poor yielder. It didn't work out very well, but then I back crossed it to my butternut project, and then it did great. And it's really interesting because that one actually has excellent keeping quality. The Chinese pumpkin doesn't have good keeping quality, and my butternut has sort of moderate keeping quality, but the cross of those two ended up having excellent keeping quality. It just, there's another story where the F1 generation of a cross had sort of moderate eating quality, and then for some reason most of the F2 plants had just like incredible eating quality, and I didn't know why. Just kind of keeps throwing stuff that I don't expect, which is exciting. And I'm excited about many of these new crosses that I've been doing, especially for keeping quality, for determinacy, and for different flavors. This is a breeding trial of something that has 25% of Johnny's butternut crossed into it, and then selected over a number of years, crossed with my variety, South Anna. Has really good eating quality, smaller size, it's productive, but not a great keeper. I put it out there this year as butternut 200 because I wasn't sure it was good enough for a real name quite yet. I just wanted to look at it a little more, but I wanted to share it anyway, get started sharing it. This is the South Anna variety. You can see it's kind of variable on size and shape. And that's because I wasn't really focusing so much on that. I was focusing on, I wanted fruits that were some kind of butternut shape, but then I was more focusing on disease resistance and flavor and yield. And yeah, this is a picture of a Halloween pumpkin project I'm working on. It's a pumpkin from New England crossed with Mexican variety. I did sort of a screen of a number of Cucurbit pepos from, I tried to find ones that were probably from more humid coastal areas of Mexico and Central America. And one of them called Tutume did really well in the late season, and I crossed it to this pumpkin from New England. So I'm working on that. And see, yeah, I think I'll leave it there. I was going to talk a little bit about bacterial wilt resistant selection. There are other things besides downy mildew that I look at and care about, and I think that I consciously care about. But then there's ones that just by selecting them in organic conditions, I'm probably getting stuff that's pretty overall resilient for our conditions, like a little bit of squash, some squash bug resistance and stuff like that. This last picture is a bacterial wilt cucumber trial. So I would love to see more work done on that as well. I did a couple trials, and it's kind of elusive, but I think that my variety has pretty good, it holds up to it in the field. It can still get it, but yeah.
I don't think we can hear you, Rachel.
Is that the end of
Yeah.
the presentation that you? Okay. I didn't want to jump in and declare the Q&A if you still had other things you wanted to share.
Yes, yes. Please lead us, Rachel.
Wonderful. Thank you both for the presentation. So we'll have about half an hour for a Q&A, and either if you'd like to drop a question in the chat, I can ask it for you, or if you'd like to raise your hand and let me know that you'd like to ask a question in person, we can do it that way too. So Julia asks in the chat, Michael, can you define market class for us? It's a term you used.
Yeah, and so there's probably some better horticulturalist definitions out there, but you can think of it as the group, the category something would be sold in, like as commodities. So like you see butternut squash, acorn squash, delicata. You might have flint corn, dent corn, sweet corn, many others. So is whatever, there's different categories that you might identify something being in. So whatever category you would see for a plant. Why that tends to matter is, like, say you have zucchinis. Once you are into zucchinis and improving zucchinis, you're going to tend to be crossing the plants that are most working within zucchinis, crossing zucchinis to zucchinis. When you have, my colleague Jim Myers talks about stringless snap peas or snap peas. There's a lot of genes that make snap pea succulent and edible that like garden pea, shelling pea, dry pea certainly lacks. And so if you're wanting to make progress, you're typically thinking about crossing a snap pea with another snap pea. If you're feeling adventurous, you cross it to a snow pea. So you're working in a very narrow space, and it kind of takes you on the downward genetic spiral where everything gets more and more similar. Potatoes are the complete exception to a lot of things there. Yeah, so it's just kind of that, if you think about what you would label it as to buy it or sell it, how you'd group it, that's a market class. And that's why they're kind of get so genetically coalesced. And then if you're looking for a variation, it's between things you would assign in different groups. Yeah. And yeah, have some more to wander around thoughts there, but I think there's a question for Edmund.
Yeah, Edmund J asks about the training techniques that you mentioned with the cucumber photo that you showed, and what sort of spacing you use between the plants.
Yeah, so cucumbers, if I'm training, like if I'm looking at individual plants, I'll plant them about 2.5 feet apart, and at 2.5 feet in the row, with the rows about 6 feet apart. So that's, it's a good bit wider spacing than you would normally use. And then for the squashes, it'll be more like 6 or 7 or 8 feet apart in the row, and then 10 feet between rows. Sometimes I wonder if I've been, so there's can be some downsides to training, especially with squash. I've noticed that like more incidents of sunburn with squash that's been trained. So you might reject plants that, that have sunburned fruit, but that wouldn't have happened if you didn't train it. I think you can address that by training more often. So you want to do it twice a week when it's busy, and if you let it go longer, you might end up with problems. And sometimes I wonder about, I have done grow outs where I trace the squash where I let them intertwine and then trace the squash back. And I think there's some strength to that. I actually want to do more of that. I'm not going to get so much of a downy mildew read on the leaves, but I'll get to look at the fruit and the productivity and taste it. And it's more work tracing the fruit, but it's less work training.
And we one of the big things when we go to the plant breeding spacing versus production spacing, and production spacing, the canopy so beautifully blots out all the weed pressure. Once you go to these wide spacings, you're just, if you want to tell the vines apart, you have a lot of daylight for weeds to come through. We've done things like rolling round bales of straw through the fields to be a nice mat when we're trying to keep it organic. There's sometimes when we can't, we're on the conventional side of our fields where they do herbicides there. But we're always trying to do it better. And so one of the things we're starting to do is you can get other squash you can tell apart, other cucurbits like that West Indian gherkin I think Edmund had. So we're starting to use those as the separators between plants. We're putting rows of a bush squash you want to grow, or when you go to do on farm work, there's ways to set up different borders, so you're still keeping the neighbor competition pressure to see how stuff does when it's not just the only squash you can see, but still make it so we have a prayer of finding them, telling them apart. Our days where we find a fruit we like and we tug on it and look around within like a 20-foot radius to see where the earth moves, that's not our most favorite harvest days.
Julia, I see your hand, but Craig has a question that I think goes with what we're talking about right now. So maybe we can hold for just a second on your question. Craig asks, is pruning part of the training method that you use, Edmund or Michael?
I haven't done that, but I definitely think you could. I just haven't done it.
It's not something we do intentionally. But sometimes the tractors or stuff has to get through. One of the things that we know as we are doing it that I know we're probably missing is a lot of these crops, the cucurbits, they will put down these secondary roots. So they love making roots. And whenever the nodes get kind of a little buried, they'll be trying to make some roots out. They're almost always trying to make some roots, these little white dots emerging. So so for weed control, sometimes we're moving and shifting the vines. And so I think there's things, I know there's things I'm missing when I'm doing that. One I hear about, so we don't have squash vine borer pressure yet. I'm sure it's coming. Edmund has a lot trickier growing environment than we do. We have like a lot more and a lot colder winter to be a nice, beautiful reset on everything. Makes it easy. But like when you have vine borers coming in and stuff that like girdles the base of the plant, like really bothers the base, some varieties can keep growing through that. And I think it's probably related to how much other roots they're kind of putting down to help feed some water and into the system.
Julia?
Yeah, do you guys ever intentionally add stress to increase the selection pressure, like drought or introducing other diseases?
The main stress that I add is just planting late when there's more downy mildew. So like my cucumber selections, I'll plant them like in July.
Yeah, and we do some, inoculate with some pathogens in the greenhouse sometimes. We try not to add any outside. So like when we're working with a fungus or a virus in the greenhouse, we're working with ones that are already around, but I'm trying to wrap up in the early spring when it's still freezing weather and it can't, if it blows out the greenhouse vents, it won't establish or kind of get in the area a little earlier. For cucumber beetle, we're usually trying to moderate the stress. We're in a space where we want most of the plants to live and look good, and then to be able to whittle it down further. In some cases, we'd like most of the plants to hurry up and die, and we can just pick the fittest. With cucumber beetles and cucumbers, with some of the types that can be bitter, it'll really show up if the cucumber beetles get after them. We are starting to do some drought work in some of our high tunnels. So we're just starting to do that because where I am, there's a lot of growers still don't have irrigation for much of the season because they just count on the rain. And so when the rain doesn't come, they're running around trying to drill wells or, and then the next couple of years won't be an issue. So it's like this every few year disaster. So yeah, the Gothic NRCS type high tunnels are fantastic for drought screens. The Caterpillar tunnels, not so much, but NRCS, those run really, really desert-like.
Jack?
Good evening, friends. Can you tell me how open source fits in everything that you're doing? Are you planning on pledging any of the varieties you're developing?
I could go. So my butternut squash lines are pledged. I pledged them several years ago. And then I keep making crosses off it. So that's the South Anna and the Butternut 200, and there's more that are coming. And I'm definitely excited to be part of it. Part of a movement that is, working on countering corporate control of seeds. And I think that this is one tactic. And I'm definitely interested in other, I don't think it answers everything, but it's one way to address it. And keep stuff from getting privatized by corporations, stuff that I put out there. So I'm excited about that. I'm trying to decide whether to pledge my cucumbers. And I have some, I think my biggest thought on it is, I almost wish the pledge was shorter, that it was something that you could say orally. I have this hesitation around asking a pledge to accompany the seeds, forever, indefinitely. In terms of just, I believe that these seeds will outlast our legal system and our language, and their these seeds and their descendants. And so I have this hesitation around saying, like, this written pledge is going to always go with the descendants of these seeds. So that's something I'm trying to work through. But I think that, as a tactic, and that I really appreciate the Aussie pledge, and I'm glad to be part of it. And I'm just thinking about this question.
Yeah, and I joined the Aussie board as I wanted to explore with what it meant more, because around the time that utility patents started happening to plants, Cornell started using material transfer agreements. There's actually out of vegetable breeding is where the genesis of it was. My predecessor started doing it. And in part, one of the things that was going on is using contract law to be able to prevent like ownership claims or privatization, or it was using some of the paperwork in a defensive way. It means that all this paperwork has to follow the seed, so every time it changes hands, the people are directed back to Cornell. And there's reasons when that makes a lot of sense, with stock seed, with a variety being the same out in the world, etc., for us to be able to make sure they understand the expectations of non-patenting, etc. Not every breeder at Cornell approaches it the same way, but this is how we do. That the system seemed to be serving us really well, up until I started working with freelance plant breeders, participatory plant breeders that were trading seed with each other. When we sent seed to seed companies, they're seldom the major seed companies, they're seldom really just getting together at seed swaps and sending seed around. So that hadn't been a concern, but as I'm working with the different groups, people with about a family initiative in Canada, the Cassie seed system. There's a lot of groups in Ontario, especially. Hi, all of you. Is many to know right now is, and so as they have like the group project approach to plant breeding, community plant breeding, our kind of tracing every seed and accounting where it is, monitoring it essentially, wasn't really the best fit. There's a lot of parallels and similarities. I think an opportunity to harmonize what I was doing at the university with Aussie, if we learned more and kind of explain to the university what was going on and figure out. So I also have questions about like what does it mean to restrict? So when we say you won't restrict, exactly what does that mean? So how can we, if we can figure out what everybody's intentions are and write that down, then that I think helps the Aussie seed be Aussie seed, but also helps us understand how other people can really understand it more and maybe participate, even if they hadn't been in an institution that would have considered it before.
Carol, it looks like you have your hand up. Oh, I'm afraid Carol, I can't unmute you. If you could press *6, that'll unmute you.
Okay, I have some basic questions about the biology of the powdery mildew. You apparently have problems with it, or excuse me, downy mildew.
But I can put powdery mildew too if you want.
Let's stick with downy mildew. The downy mildew, is it the same lines that are on the squashes or on the cucumbers, for example, and the same lines that are on different squash species? Or is it different lines?
So there's two major clades, two genetic groups of cucurbit downy mildew, the pathologists tell me. We've seen it happen where one affects is the most symptomatic on cucumber and the other is the most symptomatic on squash, or is more symptomatic on squash than the other. So there's kind of the cucumber and everything else, and the squash, cucurbita, and everything else. So they cross over, but they're distinct. And so I normally get the cucumber strain. We don't usually, it varies whether the squash strain will be in our area, but when it is, the squash get whacked. I think in Edmund, sees this squash more reliably affected by downy mildew.
Definitely.
Yeah. So it varies a little bit based on season, which one's going to blow in. It also, there's very little facts about where it overwinters. Probably in the Florida Keys where it doesn't frost and there's always some wild cucurbits is a pretty good bet. How it then shows up also in Michigan and the Great Lakes, also really early on. It's unknown how it gets there. There's a lot of finger pointing how it gets there. But so there's some dynamics of what's going on that it's really, it'd be so helpful to be able to know what's going on, so that we could have some more targeted efforts.
We don't, as far as I know, we don't have downy mildew on cucumbers or squash here in the Maritime Northwest.
Yeah.
Is that right?
Yeah.
And are you expecting that we're going to have it?
I get we have customers from California, parts of California that get it, that order from us.
Yeah. Then starting to pop up. There's a website. It's a long website, but it maps the movement of cucurbit downy mildew across the forecast. You can hear when it's nearby, when it's, I guess, to jump to your county, so you can see where it is in the U.S. Globally, it's also an issue, but not so much the mapping tracking effort. But it's the East Coast, the Gulf Coast, and then some like southern, like halfway up California is the highest I've seen it, and then the Great Lakes. But it's I think the Pacific Northwest, like when hops downy mildew showed up in the Northeast, that we lost that industry, and that's when it moved to the Pacific Northwest as well. You need that alternating really wet and dry, so that's what it needs to complete its life cycle. So I think Corvallis is probably safe for the foreseeable future.
Okay, thank you.
Sure. Yeah. And to follow up on a notion from Edmund a while ago about my crazy graphs. So I think, so if you're a freelance, or if you're interested in crossing squashes, so what do you do with that? So what we're going to be having hopefully really soon is kind of the information about which different genetic groups, like what's the family tree of squash and what squash are in different groups. And if you have one that's not there that you would like us to help figure out which group it's in, we'll be able to be much faster at that. There's some ayote, for example, that wasn't in our screen to our knowledge. So if you're interested in where that was placed, and there's a lot going on with that in a lot of ways, but to which group. But by us being able to, you can approximate things with geography and which commodity group you would put it in. But if you like some more insight, we could help you with some of these lists know exactly what's what. And so if you wanted to either have a cross where you would have the best fertility and the least issues down the road, this could help you. But if you wanted to really jazz stuff up and look for some hybrid vigor, our crosses between continents, especially, and where you have very different culinary uses, et cetera, for the squash, that's where we see some of these fantastic things. Like Edmund, that you was talking about, how you take two poor storing things and cross them together, and then suddenly, like, eureka. We see that too, and it always turns out it's between these groups. So we'll have some lists. Can help people navigate the family tree a little bit if they want. So I think that's kind of the take home from that for a lot of folks. It also reminds me of, I had Jay, I think has a relevant thing before I move on to my next ramble.
Yeah, I was just curious, using that information that you're talking about, and in that case of when you're trying to do these, relatively wide crosses, do you feel, or do you tend to want to make that cross and then start selfing, or do you feel like you can end up getting more interesting recombination between these things by sibbing for a few generations, or does it really matter?
Yeah, so depends how hurried you are, and how uniform you want it to be. So rule of thumb, in any recombinations happening between the squash, any plant, you get 2 or 3 crossovers per chromosome. So you're really cutting the deck, you're not shuffling the deck. So the more times you cut the deck, the more times it mixes it up. Squash has these 20 little dinky chromosomes, so I think they might behave a little differently. But in general, you don't get many crossovers, and squash is less, if anything. So, and I think, so if you want to really mix things up, sibbing beyond the F1, like selfing the F1 and sibbing the F1 is usually pretty similar. You know, if you're working with a land race, like, you know, where you have variation between plants, like by all means start sibbing, and then you'll be getting more variation that way. But it's like the generation after that, you know, keep sibbing to really stir the pot. And, you know, there's different philosophies about if you want to be stirring for a while, sibbing for a few generations with patience that I lack, and then look to see everything that's there. There's reasons to do that. You retain more things you didn't see, but then my goodness, you have a huge tangle to untangle. Someone like me would like to look at F2, find my best plants, self those, and then at the F3 start combining them together. I like to reduce the complexity of the puzzle. At the same time, I'm prematurely jettisoning a lot of stuff that would be really useful down the road. So there it becomes kind of like your scale and philosophy. But yeah, sibbing, if you want to really mix things up and see what all the possible combinations are, for sure. An observation I haven't really followed up on a lot, but it was the thought of Henry Munger, 2 generations before me, is that if you kind of wait too long to try to get recombination, you start to get regions that are homozygous, and you tend to have recombination happen in regions that are the same. So after you self something and then you want to cross it or sib it again and try to get more variation, the thought then was the recombination would happen in the regions that zippered up the best, the regions that were the most similar. So you'd be kind of just spinning your wheels if you waited too long. That would be really cool to keep on the genomes and see how much of that really is working. But you know, that was his insight, and darned if he wasn't an insightful person.
Cool, thanks.
Michael, do you mean like if you waited too long to start sibbing, like if you selfed too much, you would run into problems?
Yeah, and Henry was the one of his great contributions was he was a champion of back crossing. So for him, he would be interested in recovering type a lot, and so he had a technique that gets a lot of eye rolls around, but I still have used it from time to time, and I've told myself it's made all the difference. We could probably look at some point and see, but it made sense to me. So he had a technique he called the half back cross. And so rather than crossing back to the plant he wanted it to be like, you know, if you're working with something that's really bitter and spiny and et cetera, you want to purge a lot of that. And so when you're doing back crosses, he was feeling that he was having some of these issues of recovering type. And so he would cross things back to the F1 instead of to the favorite parent. And there's some cool things of thinking about how things are still very dynamic. You're still keeping some progress, and so keeping a lot of things dynamic. So encouraging crossing overs, being kind of random versus patchy and unproductive. And also trying to it has an element of recurrent selection still to it, even though you're trying to recover type. So the half back cross, there's a couple of essays he wrote. I think it appears in, otherwise it does not appear in any genetics text. And that was a decision of the authors, I'm sure. However, if you're looking from generational insight, there are some gems. Yeah. Oh, one quick little ramble that reminds me of. So in cucumber, this Bill Holdsworth, one of the students behind a lot of things, but even those Downy Mildew cucumbers did a lot of work and the recurrent selection that I wouldn't have done, but Bill convinced me was the way to go, and he was right too, for stacking lots of little genes. But one thing that Bill's the defense seminar that stuck with me, he was talking about how it was so much work to try to take the downy mildew resistance from these wild sources in India and bring them into like a good cucumber, like the target cucumber. And thinking about that thought, that he shared at that moment is affected a lot of my thinking. So in one, I spent a lot of time thinking about like what makes something good. And what makes it so you'd want to try to recover something. So why is recovering type so important? And the resistant types tend to be the Suyo long types. Edmund has showed one of those. There's also some Puna Kira types. So Puna Kira, so it'd be Kira is cucumber. Pune is a big place in India. So there must be a lot of people that like that cucumber for it to be the cucumber of Pune. So some thoughts about, so what really is market class? How is that holding us back? I like to wax poetic around that. I won't give you the long version here, but it shaped a lot of my thoughts. And the other is, that was kind of one of our first insights of bringing together really different genetics. Edmund is bringing together some really different genetics. That homemade pickles, a lot of pickling types had some intensive breeding for downy mildew resistance. They used a lot of Henry Munger's Market Moore poinsettia work. So I think that explains why there's some cool genetics left over in there. And then like I was doing with the Market Moore in 97 is combining it with some very different genetics from another part of the world. And so it's the additive nature of bringing together these very distant groups, things that you wouldn't consider the same cucumber, gets us, that is the key to my downy mildew resistance breeding. I don't know to what extent Edmund also finds that's the case, but that seems to be a recurring theme now in a lot of what we do, and we're trying to see like how much we can reproduce that, like in some of the other crops we work with.
Yeah, I'm definitely interested in casting a broad net to find resistance sources, and like my cucumbers have a parent from China, a parent from the Philippines, that I think, yeah, I had a really broad diversity of shapes of fruit coming from it and colors even. And I've actually had this new, a cross that I'm excited to start working with. I kind of refound it, but it's a Puna Kira type cross that did well on our trial, crossed to the same Chinese cucumber parent that I have in the other population. I'm excited to just see what happens if I just cross those and don't use an American cucumber with it at all, because it kind of seems to me like somewhere in the middle between them is kind of similar to an American cucumber. So I want to check that out. And I guess the other thing I wanted to say, in terms of like the diversity, like crossing really diverse kinds of squash, like sometimes I've done it and gotten bad results, so you just try different things, and some of them are going to be great.
We have between the Halloween pumpkin group and like acorn squash. So those crosses, I think there's a lot of cool things to be done. You end up after a couple of generations, sometimes with a surprising amount of sterility issues. Wow. So I guess when Rachel was still with us, there was some people out in the fields doing some pollinations, and they were having to use toothpicks to try to scrape some pollen out of the flowers, and it wasn't pretty. And so yeah, so those were always doing well. Rachel's acorn by delicata crosses, etc., where it was within a group, and wasn't as much genetic variation, but there was a lot more forward progress of the things that worked and stayed working. And so
well, if we don't have any more questions, I'd like to take the time to thank everyone for joining us, for sharing your time. And thank you to Edmund and to Michael for the presentation. The recording of this webinar will be available on the OSSI YouTube channel in a few weeks, along with a link to the transcript. And the April webinar on the Dwarf Tomato Project is up there now for anybody who wasn't able to join that. We'd love to hear from you what you thought of the webinar, and if you'd like to share feedback with us, you can email osseedinitiative@gmail.com. The next webinar in the series will take place on June 8th at 7 p.m. Eastern Time. Dr. Deppy will present on breeding for climate change, regional adaptation, and wild weather. So thanks again, everyone, for joining, and I hope you have a great evening.
Thank you, Rachel. Thank you, Michael.
Thanks, everybody. It was good to talk, Edmund.